And actually, before I start this presentation, I want, which I didn't do, didn't do my first, and I take a chance to do it now, to thank Barnos especially and all other organizers for inviting me here, because it was my long promise to come here to her. And now finally I have a chance to keep my promise and be here and present here. This is a big honor for me, and thank you very much for this opportunity. And now I would like to present my second talk. And my second talk, I will speak English because I'm a little bit tired. So, and in time-wise, it will be easier for everyone. And now I will talk a little bit about my own research on it. I will introduce myself, and I will tell you what kind of work I have done in Brazil, because now I'm working in University Halle-Wittenberg and leading the plant transformation genome editing facility. But before, I had a very beautiful and productive years in Brazil, working in genomic research center for the climate change, and in Campinas, in the University of Campinas. And this, I think it deserves to be presented, because this is a true biotechnology. It would be nice to have this clicker, right? How to change the slides? I don't see it. Где эта штучка? And so this talk, I will tell you about the Maize biotechnology and how it was developed during my work there, and what we could achieve, and how it can be used. Clicker. Где? So, yeah. Oops. What have I done? Что мне делать сейчас? Вот все. Okay. So I had my own journey, because it is kind of, because Anastasia was presenting our work in Russia. Now Johan was presenting our work in Germany. Now I will presenting our work in Brazil. So I will explain a little bit how it's all connected. And so first, so the journey starts in Siberia, when we actually live in a quite cold climate, as Anastasia explained, and we deal with this cold problem. And then we also collaborate a lot with famous Vavilov Institute, the first gene bank. And we have some project with the Vavilov Gene Bank in St. Petersburg, and in Germany. We had some barley-related genome editing projects. And in Brazil, we deal with quite opposite. So we deal with extreme heat, and not cold resistant, but extreme heat resistant. So it was quite interesting to travel across the world to see all these challenges and approaches. So as Anastasia showed, it started in Novosibirsk. I will not tell about that, because she did it enough. And that was a group which now I left this leading position of the group, but the group stays and the projects are running still. And then we had a very good collaboration, so lots of joint project with Vavilov Institute, and we tried to use the germplasm potential for some project. Also, it was wild potato project, and our wild potato collection, it originates from Vavilov Institute in St. Petersburg. And this is a very wonderful and very special place, and it's a great, let's say, it's a really great experience to touch this initial, like first gene bank materials. It's very cool to work with gene bank collections and try to establish biotechnology for them. So we got some experience in that. And it's really useful not only for potato, we can actually transfer these approaches for different other germplasm, including maize. And of course, this Gerasimova was is our long-lasting collaboration partners and PRB group from Johan. So we did lots of study on barley genetics together. And finally, my journey was, so before I came back to Germany, I was working like around 3 years in Campinas. And this was a like freshly established biotechnological lab or hub. It was Genomic for Climate Change Research Center. And it was, of course, focused mainly on the maize because Brazil is one of the most, the biggest maize suppliers in the world. And here it's shown that the yield and the area in Brazil is permanently growing under the field of maize. And here you can see how the maize is distributed in Brazil. And the most of the maize is produced in the actual very tropical areas. That's why the tropical climate with its very specific conditions is so giving their own challenges. And this tropical maize production is actually very interesting for biotechnology because I will tell a little bit later about that. So this is what actually, yeah, this is how it was developing. And this is a little bit about the place where I was working. So the Unicamp is University of Campinas is a I think it's the second university in Brazil and one of the first in South America, and it has a huge campus, including the huge biotechnology campus. And it was a great experience for me because Brazil, all the research in Brazil is not like Johan just mentioned, like shelf-oriented, but it's really market-oriented and field-oriented. So they only aim to go to the field and develop the true solutions for any challenges. So they probably don't do much sophisticated research, but they really go to the end from trade to field to the gene discovery and to commercialize it. So it was very great experience to be part of it. And this is a big hub having like some companies, some genomic sequence facilities, the field, which is also important, and of course, molecular labs and plant transformation facility. So the GCCRC itself was built as a joint lab between a biotechnological government company and university. And of course, it collaborates with lots of both academic and commercial institutions. And so the aim of the center was to develop a solution. So they were developing the microbiomes product, and they were applying genomics to find some stress-resistant genes. And of course, so I was part of establishing the full cycle pipeline for production of transformed and edited maize in wide diversity of genotypes. So actually, this challenge of genotype dependence is the biggest one in biotechnology because you can do whatever you want in your variety you like, or it's your model variety, but actually the true thing they require the elite varieties or the breeding donors, whatever. So, but not so this is actually a challenge, and we must establish that. Okay, and this is actually this is the lab. So it has a lab. It has a plant transformation building. It has a building for plant with plant growth chambers, greenhouse, and so field is not it's not shown here, but field is at last point. So it starts from lab and goes then to plant transformation, to greenhouse, and then it comes to the field. And so the center was established like in 2018, I think, and I joined in 2022, in 2024. So it means I was kind of part of the whole story. So they started from scratch. Even they built this a small building. At the first, they established, this is the team, they established the standard MAIZE protocol from 2019. So it was established in 2019. It was not very good. So even in B104, which is variety of this is model variety for plant for maize transformation. It was not very efficient. But then after I came, or at the same time when I came, they also we also already established this morphogenic regulator genes. We used the Wucha, the baby boom genes for transformation, and this was really helpful in a sense of efficiency and overcoming genotype dependency of transformation. So this is a little bit of statistics of our transformations. So here, how many embryos were transformed every year? So 2020 is drop, I guess, because of the COVID time, as you can imagine. And how many plants were regenerated? And actually, you see that it is just thousands of embryos and probably like single units of plants. It's not that many. And actually, this is regenerated plants and transformed embryos in the periods of the high and low temperature seasons. And you can see that in the high temperature seasons, like in summer seasons, it's going way better. And in winter season, the regeneration doesn't go very well. And when you look here, here they show the efficiency of let's say embryo formation. It means how many days. Um, so the days from pollination. In how many days embryos reach certain shape or something like that. And you can see then in winter time, like June, July, August, which is actually summertime here, but it's winter time Brazil, embryos require way longer to maturate to the stage, this optimal stage for transformation. And the red line, this is where we can experimentally define the best stage for embryo or to isolation, because if we isolate embryo when they waited too long, they will not be transformed. And actually, this was a critical point. So we were not capable of transforming in the winter months at all, but because embryos were not reaching the size in a certain number of days, and if they don't grow, they cannot be transformed. And here also you can see the efficiency of transformations between the months. It's also interesting when you see that June and July is not giving much, with some probably, but if you see, for example, some other like April, May, or March, it's much better. So somehow we realized that July and June would not be the best seasons, and actually after some time we even stopped pipeline for in the winter season, because it was like a huge work but with like zero outcome. But in summer seasons, so the transformation worked. This was the situation till 2022. By 2023, we actually started to transform with morphogenic regulators, which is Woodford and Baby Boom, and the efficiency jumped many times, actually. So as soon as we started using this Baby Boom Woodford genes, which help transformation, most of these problems were kind of done. So you can see this became way more efficient, I would say. And here is a number of transgenic plants which we were capable to obtain without using morphogenic regulator genes and using morphogenic regulator genes. And this is really improving the story. And but still, so still in the winter seasons, it didn't work very well. So I would say so. But generally, if we take overall transgenic plants obtained without morphogenic regulator and with morphogenic regulator, we see enormous, like it's like more than 10 times more efficiency, which actually proved that the method is working, and then we can even applied for solving different scientific tasks. Yeah, and here is the efficiencies for the 2023 when it started working, and yeah, some transformations were still bad, but overall, so we started to get better and better results. And this is also interesting to show. This is so the transformation efficiency in Maize is highly temperature dependent, is like protocol dependent, and but also genotype dependent. This I will talk a little bit later. But what is interesting is also dependent on which year we take. So actually, we found out that if so, we always mark so which year we take. For example, we use 7 years in a transformation, but then. So sorry. It appears then, for example, if you look at the experiment number 56, 1 year gave us at the end like 10 plants, but none of others did gave any. So it means that transformation efficiency is this mystery which doesn't only is not only dependent on something. So it's dependent also on spike itself, but all the spikes, they look the same. But when you really keep track of all the efficiencies, you see that one spike produces transgenic plants and another plant spike doesn't produce. So this is a mystery, and we don't know actually what is the response, because we try to do the same size of embryos and so on, but just what year we take, yeah, we don't know how to improve the quality of years. And the last transformation method, which was currently establishing there, and I was also part of in the beginning, it's leaf transformations. It's freshly published, relatively freshly published method, which allows you to transform young leaf without isolating embryos. And it is pretty promising because we don't need even a greenhouse to produce our donor material. We just need some petri dishes and some seeds. So we germinate them, then we apply the protocol, and we have transgenic plants. Of course, it facilitates a lot the process and also makes it way cheaper because you don't need to maintain a greenhouse, and of course faster. So this methodology gives lots of promises. It's also pretty much dependent on everything, like for example, agrobacterium strain and vector and so on. But I think so. Here you see the plants which already regenerated from leaf transformation, and we are waiting for the progeny to see whether they're transgenic or not, because it looks very good and produces plant in a very short time if we compare it with our normal protocol, embryo-based protocol, because as you know, so every company highly interested in reducing cost and time, and this could be a very nice solution actually. And when I came, I also decided to add to the system, I decided to add this new, relatively new ternary system. So normally agrobacteria, as I mentioned in my first talk, it consists, it has so 2 vectors. One vector is TI plasmid with Vir gene, and another vector is our binary vector we use for transformation. But ternary system, it has 3 vectors, and the third vector is just packed with the Vir genes. And here you see some transient expression test for binary and ternary system. And for the binary, the standard agrobacterial system, you see some dots on embryos. And when you use ternary, the whole surface of embryo is transfected. And this is cool. And so I decided to apply this to the tropical maize to look how it's going to work. So we applied both actually this Wushel baby boom morphogenic gene assisted transformation plus ternary vector, which is very efficient in introspection. And we can see that the control doesn't go because here is not is poorly regenerating tropical maize. It is not because B104 would generate in any condition, but if you take some genotype which is very hard to transform, and then we apply this morphogenic assistance to help to regenerate, and the ternary system to boost the transformation efficiency, we see pretty beautiful embryo structures coming out from embryo, even after 1 week, you see these bubbles, and then they growing, and then they go to regenerate, and they produce very nice plants. And so this is how it looks like. So if we mark it with some reporter gene, you see that these bubbles, they normally express your reporter. And after establishing the transformation pipeline, also you know that it's not enough. So if we have transformation plants after transformation, we also need immediately to see whether it worked or not. So that's why the phenotyping pipeline is highly necessary just next to the transformation pipeline. And so we established quite a simple version of phenotyping. They were just taking pictures and then analyzing some characteristics of plants, and it gives us already the first idea, for example, in stress experiments with seedlings, about some general plant growth performance and how it looks, so actually it allows to this phenotyping allows to extract very good number of different characteristics, especially if we go for the immediately with seedlings for the some drought tolerance productivity. It can give us immediately the first idea about whether our um, um, change or modification or in this particular genotype caused any change in productivity and stress resilience. So we kind of make this plots and group the genotype in a certain not genotype, but for example, a population with screen, which differs from control in both productivity and stress resilience, and divide them into the groups with bigger productivity, less resilience, less productivity, more resilience, and so on. So this gives us the first idea how our approach works, because mostly we will focus on the stress and the heat resistance. And then, since we had field, so it's also great that we could test irrigated, non-irrigated conditions, and we used, if we had, for example, our first transgenics in B104, so not in tropical lines, because in B104 it was well established as a model. Then we cross it with specific tropical lines to make it possible to grow in a tropical conditions. And then we had this drone phenotyping from above, which could allow us to extract some indices and immediately see not so after we have the lab performance, we also could look at the field performance of our material. Well, and since all this was established, I mean, the transformation, the phenotyping, actually the another pipeline which was there. So it was a gene discovery project, because mostly we're interested in finding new genes which are responsible for stress. And it was first based on the genomic and transcriptomic approaches. And then after we have our gene candidates, so the idea was to do like overexpress and knockout lines and perform this phenotyping using our phenotyping pipeline. And then, of course, as I said, the result would not be paper. It actually must be patented and go to some product and to the market. So this is an idea. And the project itself, so in previous years, our colleagues, Ricardo and Isabel, they took extreme resistant species for the very severe conditions, and they performed some genomic and transcriptomic study of the species. And then the idea was to define the homologous genes in Bredopsis thaliana, which are not characterized at all. So first, they defined the group of genes for Bredopsis thaliana which were not characterized at all. And then among and homologous to this extremophile plants, and then they in this unknown genes, they took the genes which were co-expressed with some stress transcription factors. And then we came to some small set of genes which are unknown but highly related to the stress and to the species which are highly stress resistant. And then these genes were first analyzed in Lepidopsis thaliana, and then it was a big job to find maize orthologous genes, but also the maize orthologous genes we looked based on their behavior in different stress conditions, and then we found very clear, also the genes are not characterized, we don't know which are domains, nothing about them, but we see that they are highly they really highly respond to stress. And for these genes, selected set of the Maize genes. It was the job to do the overexpressor construct and do all these things. And we did find the very good positive effects of these genes, but they were in part, and some of them were already patented by other people. But that was a good confirmation that approach actually works. And this helps to find a new but very helpful genes in the sense of stress resistant. And here was also my job. So I joined the group at the moment when they were developing also the knockout constructs for these unknown genes. And so we could see that both overexpression and knockout are working very well in maize. And so the main topic which I want to talk, because this was kind of state of the art when I joined the group. So they had the transformation and knockouts, and then then it was a challenge to actually to come to the tropical maize, because whether we can establish the same technology for the tropical maize and modify it somehow to because tropical maize, it is, as you see here, the temperate maize is a highly productive if you compare to the tropical maize. The tropical maize, it grows in a huge area. It has like a huge, as you can see here, area, but not that high yield. So the tropical maize, it means even having very good characteristics in sense of the resistance to any severe extreme heat conditions, it is still like it has, let's say, breeding potential, because the temperate, of course, the temperate lines, they had more, the people worked more on the breeding, and actually tropical maize is very promising in the sense of coming climate change, but the biotechnology is not really established. So to start with, we decided to take a model which was provided us from the VIB, from Ghent, from group of Lawrence Powers, and they developed a very nice model system which shows you how genome editing or knockout works. So there is a so-called wheel gene, so there's a yellow-like gene, and well, it is knockout. You can see the color change, and the transformation was also established in the Lawrence group, and it had also used this Wushel, the baby boom. So we decided to apply exactly the same approach to establish the technology on the tropical maize first. So first, we as a proof of concept, we got this in V104, so it worked immediately and very well. And you see how plants are striped, because you can see the phenotype, the chimeric phenotype of this wheel genes. And the tropical maize we took from the collection of CI MMYT, International Maize Wheat Improvement Center. And we selected, is it here? We selected actually two so it was 3 tropical maize lines, and which from this collection, and also we took 2 commercial lines from our partners, and we checked the sequence of the gene. And some of the lines, they had one mismatch in a target motif, because we wanted to use exactly the same system. And what was interesting, also this target motif had a off target in another gene, which also had a phenotype. So this off target phenotype is white, the wheel phenotype is yellow, and the wild type is green. And first we did a protoplast, like it was many times mentioned here. And surprisingly, we confirmed pretty nicely mutagenesis, even in alleles with a mismatch, showing that the Cas9 system is not that precise, and the efficiency for the motif with one mismatch from the guide RNA is still pretty good. And when we came for this, so as I mentioned, it was 3 lines from collection and 2 from some commercial partners. We applying this protocol with multigenic regulators, we immediately have the striped phenotype, as you see here, and it means that we got immediately the highly chimeric T0, which is not, it's not clear how did it work because it was all like this. And actually it is showing pretty well what you get, because normally your phenotypes are not colored, but now you can see all type of the stripes, like a wild type. You can see the mutations only one gene. You can see the combination of the mutagenesis of your target and off target immediately, and we're kind of confused. And for genotyping, we applied this. So we PCR based, so we do the PCR, and then we apply this specific algorithm which splits the Sanger this chromatogram and gives you some. So it splits it automatically and defines the mutated allele which are present in these highly mosaic plants. And so we analyzed our T0 generation using this algorithm. But since we were not pretty sure about the efficiency algorithm, we also tested it with our self-made approach using deep sequencing, which Anastasia already mentioned. And luckily, we had a very beautiful correlation between both approaches. So it means that we could clearly, with not that high error, define which type of genotypes are present in our samples and which proportion. And so having everything this established, so we obtained this amount of this kind of efficiencies, the efficiency in different lines right from 0 to 6%. And you see for CML 888, 488, we didn't get any plant, but for all other lines, we could get like some indel frequencies at 97% of the T0 plants in tropical lines, and with a different. So we got everything: homozygous, heterozygous, mosaic, and biallelic plants in a different frequencies and different lines, and we also got pretty much of off-target sectorial activity, which, and by the way, as you see, off-target is different in 3 nucleotides from target site. So even having 3 nucleotides different, we can see quite enough off-target activity then. So this is the next generation, which is also worked. So we see the phenotype and genotype, it's all there. So we decided that the methodology is generally established in the tropical lines and decided to turn to the real rate. So the first, since it was very, very popular talk about the short corn, we decided first to address this trait and try to make the tropical corn shorter. So this was my project. And I decided to take as many genes as I can without even looking much at them, and try to design the multiple gene editing system, which would try to target different genes and probably so create some diversity of phenotypes in the sense of the plant height. And as a model, so we decided to take the gibberellin gibberellic mechanisms of different forms of gibberellin. Actually, there are many genes which all called gibberellin oxidases, but they're not only oxidases. There are many, actually, they are responsible for many different biochemical conversions. And I designed So I decided to take, since morphogenic regulator system worked very well and ternary system worked very well, I decided to combine the ternary system plus morphogenic gene assistant transformation to apply for modification of this tropical lines for the multiple genome editing. And we use this, this contains also baby boom butyl morphogenic genes, which and LOXP, which supposed to be cut after the transformation, and a ternary vector and multiple editing. So we got some regenerants. And actually, I want also to show you some results. As you remember, I mentioned that for CML 488, we didn't get any result using just morphogenic genes-assisted transformation using binary vectors, but with the ternary system, it worked. And also, if you see with B104, we didn't find much difference between binary and ternary system, but when we look at the other genotypes or tropical lines, we see that the combination of morphogenic regulators and the ternary system, it really helps to overcome genotype dependency of the plant transformation and genome editing, because we got some effect in any lines we tested. And these are the results. And also, according to expectation, we got a very wide diversity of the plant shapes, and including some plant here which even look more animal than plant, but unfortunately we couldn't get any progeny from this guy, but from others, yeah. And these are the lines in T1, and you can see that this like T1 seedlings, they are pretty much the same. It means we don't have that much, so it is more or less homozygous, the mutants, but the phenotype is very different. The phenotype was really different, and like uniform across our T1 progenies. That's why we decided to apply our phenotyping system, which helps us to follow how the plants were growing during the time. And also we applied some in greenhouse, some manual phenotyping, and this allowed us to extract characteristic, kind of make some, so to see what kind of phenotypes we could get, so what sort of diversity. And this graphs, for example, shows the distribution of the phenotype having two different characteristics. This is the height, like the shape of a plant is more tall or more like big, like white, or more slim. And we got this interesting B104-like variants, which are actually with a big round, the B104, the temperate line, and we got quite a lot of plants in the shape of temperate line. We also got some very strange, what as we call it, tiny maize, which was really tiny in a shape. And also we got this wonderful, and as expected, the real short maize in the tropical background. And so this tropical short maize could, you see how different it is in its shape, but it was normally producing spikes. And actually, what is interesting that the different backgrounds, they respond to the same treatment differently. So we had the same guides in different genetic background, and they produced as different phenotypes. It means that, but unfortunately, we don't know the genomes of these lines. So actually, it was kind of blind story because, but now seeing the diversity of the phenotype with the same treatment, we can assume that we kind of discover the potential of different germplasm, because different germplasm with the same treatment produces different types of shapes. And this is very interesting. So now we need to do genome sequencing to find the alleles responsible for this weird shapes. And for the plants which have mutations which we could identify in loci, so now they will go to field, because the previous, so some brief evaluation of their stress resistance showed that even having initial control plants pretty resistant to stress, but it appears that the short variants are even more stress resistant. And actually, it is what I wanted to tell you about this Brazilian story. This is a wonderful group. I had a chance to work and learn a lot there because it was the most exciting experience, because I could learn not only the different technology, but also this story, how to start from the hypothesis and come to at least to the candidate genes, which proved or very promising function. And thank you very much. So, yes. So Barno is not here. Ternary system just has another vector with lots of weird genes, nothing special. So it was kind of the collection of the weird genes put together in one vector and transformed to the strains. And different strains also respond differently, but yeah, some systems work very efficiently. Ah, yes, let's check. So, and so, yes, maize leaf, yeah. So it's somewhere in the beginning, at the beginning of establishing the story, yeah. Post transformation protocols established. Yeah, somewhere here. A little bit earlier. Ah, sorry for such a long presentation. Ah, oh, back, back. We are approaching. Here, yeah. What's focal chief? Yeah, this is a standard. So first you do the preparation of explant that you co-cultivate with agrobacteria, and then you go for the selection resting media. So you just give all the opportunity, all the process to be done. So all the genes of antibiotic resistance to express, and then you select with antibiotic, and then after selection, so you allow only to grow the selected antibiotic resistant cells. I guess this one is B104, which is, but as you see here, a little bit here, so we also tested tropical lines for that, and they respond pretty good. So tropical lines look promising in this method. Yeah. Oh, it's a good question. I am going to test this. This is my actually idea to do now, because it just, yeah, it makes donor material story like a very easy, simple. Yes. Yeah, it is. It looks very good. So it's not easy to do. I mean, it's tricky, but if as soon as you have your working protocol, so you can try, and we are going to try. Yeah. Yeah, of course. As I explained, the embryo having certain size in certain months of a year, like in summer, embryos having certain size and having a pretty well established, like the ternary system based protocol. Of course, they have very high efficiency, but for leaf transformation, this is not yet very high, but it is still in the process of optimization, I would say. I hope we can make it more efficient. So it's immature embryos of like 2 millimeters size, immature. Yeah. So if no more questions, so thank you for your attention, and I hope we can have coffee, right?
Yeah, I think we're going to close the section, today's section, and thank you everyone for that nice talk. And I wish you have a wonderful afternoon and evening, and then see you tomorrow.
Bular ya. Bular nima deyapti? Yeah, I don't understand. Yeah, it is what I yeah, that's yeah. See, yeah, oops. Laptop, second. Laptop for me. So, okay, we can project. That is get right. Yeah, it's not a policy. Okay. Now that's one part. How do you get the setting girls? You don't need a big screen. I don't want to show the vision for it. Looking to the You can stand here and controlling.
Yeah, that's usually it's better. But I can check if it's working.
Okay, okay.
Like this. But can you start off? Be checking if you can use your own computer. Okay, I just then we'll check out in case if it's not so there. Okay, then we're both asking, can you start off computers?